Hello friends, in today's video we are going to talk about a very important term which is called breakthrough curve. So usually breakthrough curve is used in column adsorption. So this is the plot of concentration ratio. Usually when we say concentration ratio, it means Ct over C naught versus time. Okay, so this is also called a dynamic plot, and this is called breakthrough curve, or sometimes you also say this as a concentration time profile, and it is obtained from a series of column adsorption experimental data. And the breakthrough curve and curve of the breakthrough graph indicates the dynamic response of the adsorbent system. Okay, so breakthrough usually takes place when the adsorbent starts to exit. The pollutants start to exit from the other side of the bed with the concentration that is equal to 5% of the initial concentration. And usually the shape of the breakthrough curve is S shaped. Okay, and in most of the cases, the absorption by the column method, the breakthrough curve exhibits a S shape, but with varying degree of steepness. So you can see here, this is a breakthrough curve, for example, and this is my Ct over C naught, and this point, okay, this point is my complete. You can say that everything has been used up, and here this is my break point, and this is my breakthrough curve, and here this is the complete exhaustion. So everything has been absorbed. Okay, now so you can say this point as also a complete saturation point, and this as a break point, and this as a also breakthrough curve or a break curve. And this x-axis usually, if you see here, starts to increase, and then here this is a break point, and this one time usually refers to the time, or you can say the volume of water that is going to be treated. Okay, and here you can see this area and this area. So this is my exhaustion point, and this is the area where, if you see, this is almost equivalent to 1, and this is initially which is 0, and it can also be called effluent volume, the x-axis. So this is also called the dynamic studies in fixed bed system. So most of the ion exchange operations, whether in laboratory or in plant scale process, are carried out in columns. So a solution will be passed to a bed of adsorbent beds. Okay, where the composition is changed by absorption, or you can say adsorption. The composition of the effluent and its change with time depend on the properties of the adsorbent or adsorbent that we are going to use, and also the composition of feed, the operating condition, the flow rate, the temperature, as you saw in my last video. We talked about the column, and this is the plot of the ratio. So C over C naught, or Ct over C naught sometimes. So it's outlet sorbate concentration upon the sorbate feed concentration versus time. So they are known as breakthrough curve. So initially, the mass of the most of the mass transfer takes place near the inlet of the bed, where the fluid first contacts the sorbent. If the solid contains no sorbate at the start, the concentration of the fluid drops exponentially to 0 before the end of the bed is reached. The concentration profile as well as a breakthrough curve is shown down, or you can see even at the top. So initially 0, and then it goes almost to the exhaustion point. Okay, the region where most of the change in concentration occurs is called the mass transfer. So this is the real behavior of the mass transfer process in fixed beds. When the axial or the radial mass transfer resistance are neglected, absorption occurs homogeneously, and this is the ideal case. In fact, mass transfer resistance can be often taken as C over C naught values of 0.05 to 0.95 unless any other recommendation is fixed. They are related to known as break point, or you can say the TB or CB, and the saturation point where almost all the adsorbents are exhausted. So in most of the cases, CS or C naught. This is the case of the wastewater treatment of highly toxic sorbents when the concentration limit reaches the limiting permissible value. Okay, now you can see here, this is my pictorial representation of the breakthrough point. So this is my real situation, this is my ideal, and this is saturation, and this is my break point, which is shown by different colors. Okay, so this is all about the breakthrough curve. In our next videos, we'll be talking about how to draw the breakthrough curve. Okay.